Okay, so in this case here, this handout, as I said, DNA replication, the process of DNA replication is a very common question on a test of explaining things in detail. So this handout here basically would cover all you need to have on that if a question come up and ask you that. So I'm just going to go over it and talk about it. Okay, you do need to know all the enzymes as stated, what they do, and in order. Okay, so let's go over that. Now, first part is this. You have DNA gyrase. Now, in the textbook, they talk about something called topoisomerase. But if you remember, in the textbook, they say topoisomerase is a class of enzyme that relieve the tension of the strain, blah, 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 blah. DNA gyrase is a type of topoisomerase. A type of topoisomerase. Okay? So DNA gyrase is a type of topoisomerase. And your DNA structure, if you remember, it is a double helix. It is a double helix. And not only it twisted together like this, it's also coiled, a lot. There's a lot of coil. So before you can even unzip it, open it up, you actually have to loosen it first to allow it to kind of come into a nice straight ladder form. So this is what DNA gyrase does. DNA gyrase moves in advance of helicase, relieves strains in the DNA molecule that are created when it's uncoiled. Without this action, the separated strain will form tight supercoil. So if it wasn't for the DNA gyrase, it will just recoil back into a big bunch. So that's a first step. The second step, you use helicase. A number of you have a zipper. Actually, I'm pretty sure most of you do have a zipper of some sort that you're wearing today. Think of your zipper as your double-stranded DNA closed. The role of the helicase is to unzip the DNA double strand. Okay? So helicase uncoils the DNA double helix and split it into the two template strains. So that will be here. Uncoils the DNA double helix, split it into two template strain. And over here, this little thingy, here, that's your helicase. I just noticed it now. So this thing here, this circle thing here, that's your helicase. Again, is to unzip that. Now, not only the helicase unzip it, you also need single-stranded binding proteins. So these things, single-stranded binding proteins, will keep the strands apart, okay? So long enough to allow template strand to be copied. Because after you unzip it, it has a natural tendency to go back and form those hydrogen bonds again. Single-stranded binding proteins keep them apart. Okay? Are we good so far? Now, at this point, the 5' to 3' thing is important. So if you look at the original strand here, so this, the one that's on the top and the one that's on the bottom here, the one on the top, the original DNA strand, the top strand is 3' to 5' here. The bottom strand is 5' to 3' here. The main enzyme for writing the new DNA section is DNA polymerase 3. Now, DNA polymerase 3 has a problem. It can only write from 5' to 3'. It can only add from 5' to 3', which in this case, well, it's not a bad thing, because for this side here, the darker strand, that's a new DNA strand. Well, if the helicase go this way, 5' to 3' is the same direction. So for this strand, the DNA polymerase 3 can just keep going the same direction as the helicase going. So in this case, this strand here will be what you call the leading strand. Okay, and think of the arrowhead here, the DNA's polymerase 3. So in the leading strand, it just moves in the same direction. Leading strand moves same direction as replication fork, which is close to the helicase. When you assume that zipper unzip, that separation, we call it the separation replication fork. Okay. Now, you might notice here, you have, even on the leading strand, there's some primers here. And on here, you don't see them labeled as primers, but the thing is this: DNA polymerase 3, the enzyme that's mainly writing out the new DNA, it can't latch on to the original strand without some sort of primer, right? Without some sort of primer. How many of you ever painted like your house or a wall or no? So primer is the first layer that you usually put on. So you need primers. And those primers are sections of RNA. Now this part here, compared to the textbook, the handout here say DNA primase. I would say in the textbook called it RNA primase. I personally think RNA primase makes more sense, since RNA primase, it adds, so the primase are just short length of RNA, acts as primer. And that allows the DNA polymerase 3, mainly 3, to bind and begin replication. So in this case here, RNA primers have been making these RNA primers. This primer here, it's also RNA, and RNA primase put that on beforehand. Earlier. Now, RNA primase work on both leading strand as well as what you call lagging strand here. For the leading strand, you really only need to do it once because you add that RNA primer, the RNA polymerase 3 practically took over and kept going. On the lagging strand, however, this is the problem because helicase goes this way, and then when you unzip it, this is 3', that's 5'. You cannot have the DNA polymerase 3 following the same direction. So basically, every time helicase unzip the original strand DNA, RNase primer would add onto the space here, and then you have DNA polymerase 3 adding on this way. So you have sections of DNA later on with some RNA primers. Okay. So this part here, DNA polymerase 3 starts a replication next to the RNA primer, adds nucleotides in 5' to 3'. Okay. It moves away from the replication fork, so this is the lagging strand. So this here is the lagging strand. And as you see, this arrowhead is like the DNA polymerase 3. So instead of going the same direction as the helicase, it's going opposite direction. Are we good? Now, because it goes opposite direction and you basically have sections of DNA and RNA combined together, so all these short sections, so these short lengths of RNA that are formed between the RNA primers on the lagging strand, they're only on the lagging strand, they're called Okazaki fragments. Okazaki fragments. Now these DNA section used to have RNA primers onto it. You cannot have the RNase primer stuck onto it permanently because you're supposed to create a double-strand DNA. What's the RNA doing there? So you do need to get rid of those RNA. So that is the job of the DNA polymerase 1. So DNA polymerase 1 will get rid of the RNA primers and replace it with the DNA. Now, after they replaced it, there's still a nick, there's still a section that's not between where the RNA primer used to be and that DNA section that's not connected yet. So you still have sections that's gap on, you still have gap on the lagging strand. A nick is left in sugar phosphate backbone where 2 nucleotides were still unconnected. And last but not least, the last enzyme in this process is DNA ligase. So DNA ligase, if you remember in the video, is that enzyme that's holding like a glue. It seals up the nick by making another sugar phosphate backbone. Okay. So to quickly sum it up, DNA gyrase go first, loosen and release the tension of the supercoiling. Helicase unzip the original double-stranded DNA. So you have 2 original ones used as templates. At this point, you have a leading strand, you have a double strand. On the leading strand, it's simple and easy because once you have the RNA primers, your DNA polymerase 3 can go exactly the same direction as the helicase, so it just keeps on going. The opposite strand here is called a lagging strand. Lagging strand, you have more things involved. You still have RNA primase, but you make primers at various sections. Once you have the RNA primers, your DNA polymerase 3 joins in the right sections of DNA. These sections of DNA are Okazaki fragments. Afterwards, DNA polymerase 1 shows up, remove those primers, replace it with DNA. Last but not least, DNA ligase will glue or connect all the sections of DNA on the lagging strand together to have one whole strand. Okay? So this is what you will need to be able to write down if I ask you a question of describe the process of DNA replication along with the roles of the enzymes involved. Any questions? Now, another thing. So I went over the rules. One way for you to really learn the process or be able to memorize things is by using that information for practice. So in this case, this handout here, you'll definitely use your notes, basically this here, as well as your textbook notes that you've taken. This one is shown on the diagram where, on the replication fork, where all these things are supposed to be. This table, you're supposed to write down the roles of the important enzymes. I think this should be DNA polymerase 3. Last here, explain the process of DNA replication on lagging strand using references on. So this is like an example of the process, but only on the lagging strand, okay? Any questions on that? So right now, work on this. I also have assigned to you guys a puzzle video. It's a very short one, 4.5 minutes, talking about the Mendelssohn and Stahl experiment. I did ask you guys to read that Mendelssohn and Stahl experiment last week. Right now, it's just a video that will explain it. I know sometimes students kind of find that a little odd because it involves isotopes and involves growing bacteria in different isotopes, and later on you're doing a centrifuge, and you're separating them by using a centrifuge. So here's a video to help explaining that. So make sure you watch that. Any other questions? So class time right now, work on this, work on the puzzle video. If you are done, then I would say work on either your lit review or your lab, okay? I'm going to be also recording the feedback for the people who still haven't, for the lab draft. So if you have a question, if you have short questions, that's great, but it's something that's very long. You might want to wait till I do it between takes, okay? Yes?
For uncontrolled variables,